Hi everyone, very quickly I just wanted to give you the fastest breakdown I can of the mechanism of action of antibiotics, which I will have more information about in the future, but for now this is just your little lowdown. So with antibiotics, which literally mean anti-life, right? They are what we call specifically toxic. So they target bacterial cells and bacterial cells only. And they are inhibiting the ability of bacterial cells to replicate, either by killing the bacterial cell itself, causing it to lyse, and if any new bacteria are created, causing their cells through puncturing their membranes to lyse as well. If a cell is lysed or broken apart, it's not going to survive and therefore cannot make copies of itself. So, if you remember your cellular biology, you might remember that everything is made up of proteins, right? So when cells split and divide, they have to not only read directions for the nucleic acid structure, the DNA structure, the RNA structure, using messenger RNA to read these instructions, make proteins out of amino acids. Then it needs to link those proteins together and form a functioning cell. So the first class of medications that were discovered, like these penicillin type medications, they work by this first mechanism, so inhibiting cell wall synthesis of bacteria. Also, we have classes of antibiotics that inhibit protein synthesis. So, again, these proteins that make up the components of cells, that make up bacterial cells. And because mostly everything besides the intracellular compartments, like the cytoplasmic areas, is made out of protein, if those proteins aren't made correctly, you're not going to get adequate replication. There's also classes of medications that inhibit the actual making or synthesis of DNA or RNA. We won't talk about them much in this lecture. We're going to focus on really the inhibition of protein synthesis and cell wall synthesis. All right, so again, this first class of medications that we discovered, or Alexander Fleming managed to discover, are these penicillin type medications. And those are things that end in cillin. So like amoxicillin would be a penicillin type medication. And these are classified in a broader category called the beta-lactams. And the way that beta-lactams work in the most simplistic of ways is by interfering with the formation of what we call peptidoglycan, which is a component really of bacterial cell wall membranes. Bacterial cell walls are not the same as human cell walls. They are not made out of phospholipids and cholesterol. Instead, they are made out of a structure called peptidoglycan, which links together. So down here we can see this. This peptidoglycan is linked together by what we call these NAG-NAM repeating subunits. And so they have to link together with peptidoglycan. So these NAG-NAM subunits are really what gives it structure so that we can hold the cell wall together. Without this cross-linking between the subunits and peptidoglycan, we essentially then will have holes. So this was what it should look like. This is what we can have it look like without proper linking. If these things aren't properly linked to one another, then there's holes in the cell wall and the cell will rupture, it will leak its contents, and then it dies. So that's what we see here in this picture. We've got these cross links that are inhibited. So in this picture, you see penicillin in green preventing these cross links from developing. And then the cell will, again, lyse, it will leak its components, it will break open, and it dies. And a cell that is dead cannot make copies of itself. So these penicillin of the beta-lactam class, they prevent this cross-linking of peptidoglycan specifically Again, because human cells do not contain peptidoglycan, we can be relatively certain that it's not going to affect human cells. That has nothing to do with its side effects, which are due to the way that we give medication, so route of administration, and also they are due to the fact that we also have, good bacteria in our body, and when we kill bacteria non-selectively, then we are going to kill our good bacteria too. When you get the overgrowth of bacteria that normally would be kept under control by the natural microbiota made out of our good bacteria, then we call that a supra-infection. It's a superimposed infection on top of a previously existing bacterial infection. So again, beta-lactams inhibit cell wall synthesis by interfering with peptidoglycan formulation. All right, you might also remember that in the formation of proteins, we have functional ribosome subunits. And those ribosomes are present on the granulated or that bumpy endoplasmic reticulum. It's not the smooth endoplasmic reticulum, it's the rough or bumpy one. And it is bumpy because on the surface of the endoplasmic reticulum are ribosomes. So ribosomes are the functional units that actually read the messenger RNA to get essentially the recipe for all of the proteins in the cell. So I want you to think of the RNA as this messenger RNA as a recipe book. And in order for those recipes to be read, they need to be fed through these subunits that link together that create a functional ribosome. And bacteria have different ribosomes than human cells do, or any animal cells. They have different names and numbers associated with those subunits. But what is the same about them is that we have to have these subunits come together, and the ribosomes stay. My kids are screaming, serenity now. Let's just ignore them. Good parenting. Anyways, we've got these two subunits that need to link together. They come together to make this functional conformation. These two subunits in bacterial cells, they're small and large. The small one is called the 30S subunit, and then the large one is the 50S. And they come together and they stay stationary on the surface of the endoplasmic reticulum. And through these subunits, we have the messenger RNA that goes through almost like a ticker tape, or if you think of papers going through a copier, that's really similar to the ribosomes. They would make up the top and bottom of the copy machine, and the paper being fed through is the messenger RNA that we will then make copies of. The copies get spit out of the copy machine. Same thing, we're going to feed through that messenger RNA through these two 30S and 50S subunits, and then hopefully new proteins come out. In good situations, in these types of drug activities, we have the interruption of this process, either these two subunits coming together or the feeding through of the messenger RNA. Okay, so again, we have two subunits. Here's our ribosome with its 50S and its 30S. We've got our messenger RNA, which is the moving part of this piece. The ribosomes are stationary. Once these subunits come together, the messenger RNA is fed through, and then we have a part of the ribosomes that read these little triplicates. Do you remember this? Like these triplicates of the mRNA, these nucleic acid structures, and those three triplicates together, that's called a codon region. You might remember that, codon, C-O-D-O-N, and one codon region equals one amino acid. So we read this messenger RNA, we take three nucleic acid things at a time, and we get one amino acid from that. Okay, so very quickly, we have two different classes of medications on here. The first are the aminoglycosides, and aminoglycosides, they generally change the shape of one of these subunits. So aminoglycosides like streptomycin, I'm just going to write it big, streptomycin, and also this is how messy my handwriting is, gentomycin, they directly change the shape of this 30S subunit here. See how it's not flat anymore? It gets all, googly looking. What that does is prevent the smooth movement of the mRNA through the ribosome subunits so that we don't get good reading of those recipes. If we cannot read it correctly, then we cannot make proteins out of it correctly. So streptomycin, gentomycin change the shape of the 30S subunit. So also we've got tetracyclines, and in addition to the aminoglycosides, which change the shape of one of the subunits, tetracycline and some other types of aminoglycosides, they physically bind to and interrupt this process of movement of mRNA through the ribosome subunit structure. So here we would have a structure coming to link an amino acid to the corresponding codon, which is again the recipe for this protein. Proteins are made out of amino acids that are linked together to make a protein or peptide chain. So we cannot get the binding of this amino acid to the mRNA because these like tetracycline drugs, they bind to the mRNA and they interrupt this process. So we only have one functional area where we can read amino acids, and we need two. So it's like having the front page of your copying you can do, but not the back page. And it has to be double-sided to make your book. So if you do not have both of those functional, then you're not going to get good proteins. Again, the way that tetracyclines work is by jamming up, this process. At least half the process is jammed up. So if you can't read it, then we cannot make adequate proteins. All right, finally, we have macrolides, which include things like azithromycin, clarithromycin, erythromycin, and they bind to the 50S subunit. Here you can see the macrolides binding here. And again, the ribosomes are stationary and the mRNA is fed through like tickets or like papers being spit out of a copy machine, and they just come through and they block this. So it's like a jam in the copier. It's like putting in a paper jam. Nothing can come out. Nothing even matters if it's being read. If there's a jam in there, nothing's going to pop out. So again, macrolides like azithromycin, so your Z-Pak, azithromycin, clarithromycin, so things that end in mycin, they change the shape, they bind to the 50S and they block this ribosome subunit so that we do not get this functional movement of the messenger RNA through the stationary ribosome structure. Lastly, I just want to go over a concept that we call spectrum activity of antibiotics and other antimicrobial or antimicrobe, so little living things, drugs. They can either be characterized as what we call narrow spectrum or broad spectrum, which is pretty much what it means. If it's a broad spectrum drug, it kills a broad spectrum of microbial substances. If it's a narrow spectrum, it kills just specific types of microbes. So here we've got two stains for bacteria. We've got gram-negative and gram-positive. And when you stain bacteria, it either is one of those. It can be gram-positive or gram-negative. There's also really specific types of microbes like the chlamydias, rickettsias, which are only killed by certain types of these antibiotics. Mycobacterium are only killed by certain types of these antibiotics. We haven't talked about really any of these today, so these eukaryotic types of things, which means they have eukaryotic cell membranes. Like we're eukaryotes. We are not prokaryotes. Bacteria are prokaryotic. So these types of pathogens we talked about today are prokaryotic. And if we're thinking about the types of spectrum activity these drug classes generally have. Again, they can either be broad or narrow spectrum. The first one we talked about were the penicillin type drugs, and penicillins are really great at targeting gram positive bacteria. There are some gram negative bacteria that they are effective against, but they're relatively narrow in their spectrum because they are really good at, again, gram positive targeting, but not necessarily all gram negative bacteria. Streptomycin, which is a type of aminoglycoside targeting the 30S subunit, is relatively good at selecting for gram-negative bacteria and also certain mycobacteria, but is not good at all at targeting gram-positive bacteria. So we would consider streptomycin another one of these more narrow spectrum, where when you start to look at erythromycin, so one of these, again, aminoglycoside type medications, but erythromycin is relatively broad in its spectrum activity. It is selective against most gram negative, all gram positive, and selective microbes like the chlamydias and rickettsias. Same thing with tetracycline. Tetracycline is a relatively wide spectrum drug. Again, same things as erythromycin, but your tetracyclines target the 30S subunit. Your sulfonamides, we did not go over their mechanism of action, but they do interfere with DNA and RNA synthesis, and they are broad in their spectrum of prokaryotic targeting, and they are also selective against an array. I made variety and array into one word, which was varay, but a variety or array of different protozoan type substances. So there you have it. Individual drugs can be characterized by their spectrum. Like here we have erythromycin and streptomycin and polymyxin. But also we just have our classes like the sulfonamides, the penicillins, which are of the beta-lactam subclass, and then our tetracycline type medications. So narrow versus broad spectrum.